muscle first. Muscle, the muscular system is the largest system in our body. And the muscles are very important structure for locomotion in mammals. Without muscles, our life would be difficult. This lecture is important because there are diseases that attack muscles. So, like muscle dystrophies. And before we talk about the physiology of muscles, let us talk about the anatomy and the microscopic appearance of muscle cells. Muscles are divided into three. You have skeletal muscles, you have cardiac muscles, you have smooth muscles. Let us begin with a skeletal muscles. For example, let us take the bicep muscles. We take it out. Then here, like here, you are going to have something like this. Supposing this is the bicep muscles attached to the bone, tied to the bones here. This is the bone, long bone. And you take one of these units, fibril, muscle fiber. And muscle fibers are the building blocks of muscles, just like neurons are the building blocks of the central nervous system. When we take one of these, it will look like this, and this is the muscle fiber. Muscle fiber. You have in the cell wall called sarcolemma, you have a system of T tubules that gets in, that connects the muscle with the extracellular matrix. You have the cell wall, sarcolemma, and again you have a mitochondria inside here. Again, and you have the myofibrils. And the myofibrils, when we take one out, we are going to have one of the myofibrils like this. The myofibrils, again, are divided into two. You have thin filament and you have thick filaments. The thick filaments are proteins called myosin. And the thin filaments are proteins called actins here. When we take this myofibril and view it under electron microscope, we are going to have a picture like this, striated mouse appearance, striation. These stria appear because of refractive indices of light. And when you look here, this is the thick filament, and this one is a thin filament. And these are myosin hooks, and these are the actins, myosin. So, and when you look here, this line, thick line in between here is called the M line. And this one here is called the Z line. The distance between the two Z lines, two adjacent two lines, is called sarcomere. This is when you are viewing under the microscope. Now, when you take this out, one of these globular proteins, you are going to have a chain of globular material like that. This chain of globular materials has three types of proteins: tropomyosin and myosin and actin And you have tropomyosin is divided into three subunits. You have troponin I, troponin T, troponin C. It is the troponin C that is responsible for muscle contraction. The muscle contraction is called excitation contraction complex. This mechanism of muscle contraction, excitation contraction complex, this muscle contraction. Again, let's look at another type of muscles, and this is the cardiac muscles. Cardiac muscles are sometimes called myocytes. The structure of cardiac muscles and skeletal muscle is almost the same. Cardiac muscles are also striated. They have actin, they have myosin and actin. They have troponin and tropomyosin. Now, when you look at these cells here, these are muscle fibers, but they are connected with a syncytial filament. And there are a lot of nuclei. And when this muscle contracts, they work in a syncytium. Again, another property of these muscles, cardiac muscles, the autonomously initiate contraction alone, without independent, irrespective of parasympathetic autonomic innervation. When you take these muscles, you cut the heart, take it out from the body, and throw it away, it will again still perform the action of beating. It will still beat. And this is the property of cardiac muscles. And when we are going to talk about how contraction occurs in muscles later on. Another type of muscle cell is the smooth muscles. The smooth muscles are divided into two. We have visceral smooth muscles, and we have multi-unit smooth muscles. The multi-unit smooth muscles are found in iris of the eye, for example, whereas the visceral smooth muscles are found in hollow organs like esophagus, like intestine, like stomach, like urinary bladder, ureter, and so on. These muscles are not striated, but when you look at the cell, this is the cell of a smooth muscle. The cell has a central nucleus, and you have these filaments here. These filaments are called dense bodies. Their function is to anchor the actin and myosin into on the cell wall. Here you have actin, you have myosin, and this is the M line, and this is the dead line, and this is the cell cytoplasm. So this is the anatomy of smooth muscle, or the cell, the building block. And this is the building block of cardiac muscle. And this is the building block of skeletal muscles. Now, when a muscle is stimulated, say for example, a stretch, my bicep muscle, for example, I stretch it. The electrical activities that will occur are very, very quick and complex. When I stretch the muscle, like that, the muscle spindle will be stimulated, and impulses in form of action potentials will go to the cable to the spinal cord, and they will be directed back to the same muscles. And again, in form of action potentials. At the neuromuscular junction, when depolarization occurs, calcium is taken out of endoplasmic reticulum in the cell here, for example. Endoplasmic reticulum inside there in the T tubules. And this calcium will go to troponin C, and it will bind to troponin C, and this binding with troponin C will disfigure the protein, and this disfiguring consume a lot of energy in form of ATP, so that the myosin hit and actin, they combine and they will slide on one another. So this sliding will shorten the muscle length, and this is called excitation contraction problem. The same mechanism occurs in cardiac muscles, but in smooth muscles a different mechanism occurs. When the muscle is stimulated, calcium combines with calmodulin-dependent enzyme, and this one will go and activate protein kinases and cyclic AMP. And on the cell membrane, a lot of protein will be phosphorylated, and this phosphorylation will result into contracting of the muscles, and you have our peristalsis, you have to urinate, and you have digestion. So this is how contraction occurs in muscles. Clinically, we have the diseases that affect the muscle weakness, like dystrophy, muscular atrophy. There is a condition, Duchenne muscular atrophy. This is a simple example. When your muscles are weak, this is not good. The muscle of breathing, for example, when they weaken, you will die. A condition, these muscles diseases occur because of mutations in one of the proteins, then anchor the active myosin in the extracellular matrix. So this is the simple thing that I wanted to share with you. Thank you very much.